Rahman. In the previous lecture, we have studied the biosynthesis of lecithin and caphalins. Now we will study the biosynthesis of phosphatidyl inositol. For the synthesis of phosphatidyl inositol, the phosphatidic acid reacts with CTP to form the CDP diacyl glycerol. In this reaction, the CTP attacks the phosphoryl group of phosphatidic acid. So, with the release of a pyrophosphate, CDP diacyl glycerol is produced. This activated form, the CDP diacyl glycerol, then react with inositol to form the phosphatidyl inositol. In this reaction, the inositol displaces the CMP and gave rise to the phosphatidyl inositol. In this diagram, we can see that the phosphatidic acid reacts with CTP, and with the release of a pyrophosphate, this phosphatidic acid and CTP gave rise to the CDP diacyl glycerol. Then this CDP diacyl glycerol reacts with inositol. This inositol displaces the CMP from this CMP from CDP diacyl glycerol, and finally the phosphatidyl inositol is synthesized. The phosphatidyl inositol is then further phosphorylated to form the phosphatidyl inositol 4,5-bisphosphate. This phosphorylation takes place in two consecutive steps by the involvement of two ATP molecules. The ATP molecules provides the phosphoryl group to this phosphatidyl inositol in two consecutive steps to form the phosphatidyl inositol bisphosphate. This phosphatidyl inositol bisphosphate is present in the plasma membranes and is involved in the signal transduction. This phosphatidyl inositol bisphosphate is hydrolyzed to diacyl glycerol and inositol trisphosphate by the action of protein kinase C. Then the diacyl glycerol and inositol trisphosphate, which are formed from the phosphatidyl inositol bisphosphate, they act as the second messenger in signal transduction. Further, the phosphatidyl inositol contains the arachidonic acid as unsaturated fatty acid at the second carbon of the glycerol. So the hydrolysis of this arachidonic acid from the second carbon of phosphatidyl glycerol is used in the synthesis of prostaglandins. Then the synthesis of phosphatidyl glycerol and cardiolipin. For the synthesis of cardiolipin, first of all, CDP diacyl glycerol is formed by the reaction of phosphatidic acid with phosphatidic acid with CTP, as in the synthesis of phosphatidyl inositol. Then this CDP diacyl glycerol combines with the glycerol 3-phosphate to form the phosphatidyl glycerol. The synthesis of phosphatidyl glycerol takes place in two steps. In the first step, the CDP diacyl glycerol combines with the hydroxyl group present at the first carbon of the glycerol to form the phosphatidyl glycerol 3-phosphate. Then the phosphate present at the third carbon of the glycerol is hydrolyzed, and then the phosphatidyl glycerol is produced. Then this phosphatidyl glycerol condenses with another molecule of CDP diacyl glycerol to produce the cardiolipin or diphosphatidylglycerol. Cardiolipin biochemically is diphosphatidylglycerol. As we know that cardiolipin is the major component of mitochondria and it is also involved in antigenic properties. Now, the formation of plasmalogens. The plasmalogens are the class of phospholipids which contain alkyl group at the first carbon of glycerol, and this alkyl group is linked to the first carbon of glycerol through the ether linkage. So they are the ether link containing phospholipids. Another important plasmalogen is the platelet activating factor. This platelet activating factor is 1-alkenyl 2-acetyl glycerol 3-phosphocholine. The synthesis of plasmalogens is from the dihydroxyacetone phosphate. For the synthesis of plasmalogen, first of all, the first carbon of dihydroxyacetone phosphate is acylated with a fatty acid to form the 1-acyl dihydroxyacetone phosphate. Then this 1-acyl dihydroxyacetone phosphate react with an alcoholic group. This alcoholic group displaces the acyl group from the dihydroxyacetone phosphate, and so the 1-alkyl dihydroxyacetone phosphate is formed. Then in the next step, the keto group of the dihydroxyacetone phosphate is reduced. So the dihydroxyacetone phosphate is converted to glycerol, and the compound formed is 1-alkyl glycerol 3-phosphate. Then another acylation takes place at the second carbon of glycerol. Second carbon of glycerol with a fatty acid. So it becomes the 1-alkyl 2-acyl glycerol 3-phosphate. Then the phosphoryl group present at the third carbon of this glycerol is hydrolyzed to give rise to the 1-alkyl 2-acyl glycerol. Then this alkyl acyl glycerol react with CDP ethanolamine to form the 1-alkyl 2-acyl glycerol 3-phosphoethanolamine. This alkyl acyl glycerol is the compound from which the ethanolamine containing plasmalogens or the platelet activating factors are produced. For the synthesis of ethanolamine containing plasmalogens, this alkyl acyl glycerol react with CDP ethanolamine to form the 1-alkyl 2-acyl glycerophosphorethanolamine. Then the alkyl group present at the first carbon of glycerol undergoes desaturation. This desaturation is catalyzed by an enzyme desaturase, that is cytochrome B5. So the alkyl group is converted to alkenyl group. And the compound formed is 1-alkyl, 1-alkenyl, 2-acyl phosphoethanolamine, which is the plasmalogen. Then the platelet activating factor is also synthesized from the 1-alkyl 2-acyl glycerol. For the synthesis of platelet activating factor, first of all the alkyl acyl glycerol react with CDP choline to form the alkyl acyl glycerol 3-phosphocholine. Then the acyl group present at the second carbon of glycerol is exchanged with an acetyl group. So the compound formed is 1-alkyl 2-acetyl glycero 3-phosphocholine, which is the platelet activating factor. As we know that the plasmalogens are present in the biomembranes in brain and muscles. The platelet activating factor has many diverse functions. It acts as a blood pressure lowering agent in very low concentrations. The platelet activating factor also activates the inflammatory cells and so mediates the acute inflammatory and hypersensitivity reactions. The platelet activating factor also aggregate platelets. In this diagram, we can see clearly that the first of all in the synthesis of plasmalogen, first of all dihydroxyacetone phosphate is acylated with a fatty acid at the first carbon, and then this 1-acyl dihydroxyacetone phosphate react with an alcoholic group which displaces the fatty acid from the first carbon to become the 1-alkyl dihydroxyacetone phosphate, and then the keto group of the dihydroxyacetone phosphate is reduced, and so the dihydroxyacetone phosphate is changed to 1-alkyl glycerol 3-phosphate, and the reducing equivalents are provided by the coenzyme NADPH. Then this 1-alkyl glycerol 3-phosphate is then acylated with another fatty acyl CoA to form the 1-alkyl 2-acyl glycerol 3-phosphate. Then the phosphate group present at this third carbon of glycerol is hydrolyzed and released as an inorganic phosphate group, and finally the 1-alkyl 2-acyl glycerol molecule is formed. This is the basic molecule from which the plasmalogens and platelet activating factors are synthesized. For the synthesis of ethanolamine plasmalogens, this 1-alkyl acyl glycerol react with CDP ethanolamine, and with the release of CMP, This alkyl acyl glycerol is converted to 1 alkyl acyl glycerophosphorethanolamine. Then the alkyl group present at the first carbon is desaturated by the enzyme desaturase cytochrome B5. So finally the 1 alkynyl 2 acyl uh, glycerol 3 phosphoethanolamine is synthesized, which is the plasmalogen. In the same way, from the 1 alkyl acyl glycerol, the platelet activating factor is synthesized. For the synthesis of platelet activating factor, this alkyl acyl glycerol react with CDP choline, and with the release of CMP, this alkyl acyl glycerol is converted to 1 alkyl 2 acyl glycerol 3 phosphocholine. Then the acyl group present at the second carbon of glycerol is replaced with acetyl group, and this acetyl group is provided by the acetyl CoA. So finally the 1 alkyl 2 acetyl glycerol 3 phosphocholine is formed, which is the platelet activating factor. So in this way, the plasmalogens are synthesized from dihydroxyacetone phosphate. Thank you.